today's lecture, we're going to discuss bacterial infection patterns. This is quite a long lecture. It's a really key concept in relationship to bacterial classification, but also bacterial biology, because depending on the different mechanisms that the bacteria has in terms of entry into the host and replication and adherence and all kinds of things that the bacteria employ in order to survive, those same qualities are what we can also use to help us classify the bacteria. So, I'm starting with this picture of the nightmare bacteria, and this was a terrific documentary that was on PBS a few months ago. And if you get the opportunity to watch it, you don't have to watch a Hitchcock movie. This one is scary enough, and it really is presenting a real case. And it's all about this unknown bacteria that no matter what they did, they couldn't seem to treat it, and the whole hospital was in a lockdown, and they were so concerned that this bacteria was going to spread. But the documentary really brings out the importance of understanding that some of these bacteria truly can be untreatable, and that given all the science that we're employing in terms of development of these antibiotic agents, these mechanisms that bacteria have to recreate themselves in new ways are enabling them to overcome whatever antibiotic agents we seem to be able to develop. So in a sense, it can be very frightening. So we're not going to really talk too much about the nightmare bacteria in today's lecture, but absolutely, if you want to be entertained and, scared a little bit, you might want to try and watch the documentary if you can. So we're back to our classification slide, and infection patterns are really, as I explained, a very important criteria to use in relationship to classifying bacteria. So in terms of pathogenesis, in terms of the ways in which bacteria cause disease, this is just an example of bacteria entering the skin, for instance, in an open wound. And the first line, or the first type of immunity, of course, that is encountered is the innate immune system, of course. And we have the complement system over here, and we have separate lectures on each of these things, so certainly you can go back and review. And then also we have innate immune cells that engage in this process called phagocytosis, where the bacterial cells are engulfed and then destroyed. But if the bacteria certainly make it beyond that level, then the macrophages produce cytokines themselves, and also then the macrophages present the bacteria, or the different shape of the bacteria, the epitope, and that stimulates production of antibodies and engages and elicits cell-mediated immunity from the T cells. So really, this is what bacteria do in relationship to eliciting host defenses. But there are other things that bacteria do as well, so we're going to discuss it in today's lecture. So in terms of the location of infections, we have obviously many ways for the bacteria to enter the body, and then also we have many places in the body for bacteria to grow and multiply. So bacteria can infect the brain, or the covering of the brain, so to speak. We can have infections in the ears. We can have eye infections, sinus infections, certainly lung infections, skin infections, upper respiratory tract infection. We can have Helicobacter pylori, a bacteria living actually in the stomach environment, which, as you can tell, is very acidic. We can have foodborne agents causing food poisoning in the digestive or in the small and large intestine. We can have urinary tract infections. We can have bacteria living in the bladder, for instance. And then we can also have sexually transmitted bacteria. So, bacteria has a lot of ways, certainly, to get into the body, and a lot of places in the body where bacteria like to grow and multiply. So in terms of the various steps of bacterial infection, certainly we have to enter the body through various portals that I just showed you. Also, a sufficient number of bacteria needs to enter and multiply, or at least enter, for instance, because if not enough bacteria enter, for instance, then we're not going to have an infection. So the bacteria then needs to adhere to host tissue. It needs to colonize, grow, and multiply. It needs to overcome host defenses, which are, as I explained earlier, quite numerous. And the bacteria need to actually survive the host immune response. And the bacteria will certainly damage the tissue both directly and indirectly, and we're going to have a long discussion about that. And then certainly the organism can progress or recover from this bacterial infection. So in terms of the portals of entry, as I explained, it can be the skin, we can have wounds, we can have respiratory tract infections, digestive tract, urinary tract. So these are places where the bacteria can enter, but they don't necessarily need to live in the tissue that they enter, although often they do. Now, the viruses are a different story. So, and on this figure here, we show both the portals of entry as well as the different types of defenses that these portals can employ. Excuse me. So one of the primary places where bacteria live and also enter is the skin. So you can see that there are quite a large variety of bacteria that live on the skin. So this figure shows proportionately where all these bacteria live, what they are, and in which location. So in terms of bacterial pathogenesis here in the respiratory tract, sometimes it's not even the bacteria itself, but the toxins that are produced by the bacteria. So here we have a bacterium entering the respiratory tract. We have cilia, of course, that move, right, that work towards moving that bacteria out. So we need to have adherence we need to have multiplication and also adherence. But now again, some of these bacteria cause disease not by the actual bacteria, but by the things that they produce. So some bacteria actually produce toxins and enzymes, and it's those toxins that actually cause cell death. And then, of course, when we have cell death and we have particles of the cell, but the other thing that happens with these diseases too is sometimes it's the immune response that is actually the disease-causing part of this bacterial infection. So here's a couple of examples. So this is a Shigella invading and multiplying inside of an intestinal epithelium. So you can see here that through the process of endocytosis, this Shigella enters the cell. It multiplies inside the cell, and then these progeny Shigella, so to speak, actually invade neighboring cells, neighboring epithelial cells. And if you know much about the epithelium, in some locations in the body, the epithelial cells are very closely aligned with one another. And so rather than the Shigella worrying about leaving a cell and going, down the road, so to speak, in the intestine, the Shigella just actually invades the surrounding cells. It just swims into the adjacent cells, so to speak, and then infects those cells. And then what you have is you have basically an abscess that forms at that location, and that's what causes the disease symptoms for Shigella. Now, Salmonella is a little different. So Salmonella gets, through endocytosis, is engulfed into the cell, and the Salmonella multiply within the vesicle that contains the Salmonella. And in particular, the Salmonella multiplies in mucosal cells, and those mucosal cells elicit an inflammatory response. And it's the inflammatory response that causes the disease. Now, sometimes the actual Salmonella can migrate through the epithelium and enter the bloodstream that way. And then you get sometimes like a septic system-wide infection. So the numbers of invading bacteria is very important. So again, you need to have sufficient numbers of bacteria in order for an infection to basically gain hold. So there are a couple of measurements that are utilized to determine whether or not a bacteria will be successful in causing disease. One of these criteria is virulence, and that's the capacity to cause disease, and it depends on the infectious dose. So this ID50 is a measurement of the number of bacteria that are needed to create at least 50% of a susceptible population. At least 50% of them will come down with the disease. And this ID50 can actually vary depending on the place in which it enters the body. Now, potency is slightly different. That potency represents the amount of bacteria it takes to cause a severe form of the disease or, potentially death. So that's the number of bacteria that are needed to basically cause mortality. And it's the amount that's needed for at least, again, 50% of a susceptible population. So in terms of looking at portal of entry, a good example is with the bacillus, the anthrax bacteria. Now remember, I guess it was in the early 2000s after 9/11, we had that anthrax scare in the mail. And in relationship to portals of entry, and it's interesting because with the skin, you don't need that many bacteria for that infection to be successful, so to speak. If you inhale bacillus, then you need more of them, and then if you ingest, you need even more. So that skin-to-skin contact, which, is certainly quite easy to achieve, is what, could potentially cause a lethal, because the anthrax bacterial infection is pretty serious. And if you remember in the early 2000s, the male people had to wear masks and gloves and so forth in handling the mail. And there was a several-week period where it was a little dicey in terms of that mail getting to anyone. So, in terms of mechanisms that the bacteria use to gain hold, to adhere and gain hold to host tissues, we have adhesins, we have ligands, remember the fimbriae, we have glycocalyx, and then we have the M protein. So these adhesins or these ligands are actual places on the bacteria that bind in a complementary way to receptors on the host cells. The glycocalyx is a particular kind of gelatinous coating that surrounds the cell. The fimbriae, as you remember from our lecture about different structures and motility, that the fimbriae are little projections. You can see it well here on the E. coli that certainly serve to hold the bacteria in place in at certain places at the host tissue. And then the M protein we're going to discuss in a couple of future slides is a particular sort of protein that's employed in terms of helping the bacteria to gain hold at a tissue level. So here's a picture of adherence, and you can see that these ligands on the pathogen have evolved, right, to fit in a complementary way to certain receptors on a host cell tissue or a host cell membrane. And it's interesting because that can also determine, for instance, where a pathogen is likely to survive and likely to grow and multiply. So this mechanism may also be able to help determine, for instance, where that bacteria is going to live in the body, because in this particular case, it's only going to live in the places in the body where these receptors exist. So here's another example. Here's the E. coli bacteria adhering to this human bladder cell. Now you think, my God, what kind of bacteria could ever live in a bladder, human bladder? Because, obviously it's very acidic, but certainly the E. coli can. So here's another picture of adherence where we have these skin bacteria that are adhering, of course, to the epithelial cells of the human skin. And now these group A, this is an M protein, okay, and beta hemolytic strep, and they're utilized in relationship to adhering to the host tissue as well. So, there are several mechanisms, there are several ways that the host has, or not the host, but the bacteria has in terms of penetrating the host defense. So some of these are capsules. You remember we have gram-positive, gram-negative bacteria, and then we also have capsules. The cell wall. What does the bacteria have in the cell wall? Does the bacteria have enzymes? If it's a gram-negative, are there enzymes out there in the outer membrane that could serve to overcome the host defense? Does the bacteria secrete enzymes? Are there enzymes in that outer layer that can serve to overcome host defenses? The antigenic variation. Now we're going to have a completely separate lecture on this topic, but the particular antigenic variation of the bacteria itself, is it able to maintain a low profile, right? Maybe it doesn't have any antigenic capacity, and then the host doesn't launch an immune attack. And of course, that's exemplified by the intestinal bacteria. And then, can and does the bacteria actually penetrate into the host cell cytoskeleton? So the capsules, you can see actually with this picture, you can see that these white capsules are actually surrounding a diplococcus, and the capsules are composed of gelatin. It's a gelatinous material that covers the cell wall of some bacteria, and sometimes these capsules, so to speak, are composed or can be called glycocalyx. They increase the virulence of the bacteria. They increase the capacity of the bacteria to cause disease. How does it do that? Well, just from this picture alone, you can see that these capsules actually provide another layer of protection. So the capsules also function to impair phagocytosis. But on the other side of the fence, the other side of the story, the host can develop antibodies against the capsule itself. So then when the capsule, then the actual immune system can recognize the capsule as being a disease agent, for instance, or even develop antibodies against the shape of the capsule. So here's another picture of the Streptococcus pneumoniae. It obviously lives in the respiratory tract, and you can see that the capsule is protecting a gram-positive bacteria. Now remember, depending on how these cells divided, right? They divided in a linear plane, and they are, two of them, the diplococci, or coccus, are actually surrounded by this gelatinous capsule, which gives it another layer of protection. So the cell walls themselves can have components that evade host defenses or actually overcome host defenses. So the M protein, for instance, is on the cell surface and also on the fimbriae, and they mediate attachment of the bacterium to the epithelial cells of the host. They also help the host resist phagocytosis. However, the host, right, it's not a win-win game here. The host can develop antibodies to this M protein as well. So here we have the fimbriae, here we have a capsule, and we have the diplococci. So certainly we can utilize the fimbriae to actually hold on to, so to speak, the host cells. And then there's another outer membrane protein called OPA. And then after the bacteria attaches to the cell, then potentially, or the host cell, then potentially the host cell can take in the bacteria. So that, now, here's another way in which the bacteria can overcome host defenses, is through these bacterial enzymes. Now, these enzymes can actually, certainly they can be proteases, they can actually work towards, digesting host cell material and facilitating invasion and even destruction. So we have coagulase, we have hyaluronidase, we have collagenase, and we have IgA protease. And depending on what the enzyme is, it can, it can hydrolyze, it can coagulate fibrinogen, it can hydrolyze polysaccharides, it can, spread gas, gangrene, and IgA proteases can actually work directly to eradicate and destroy immunoglobulin A, which of course is produced, which is an antibody or a group of antibodies, and that these enzymes themselves can work to destroy host tissue, but also certainly overcome host defenses, especially the IgA protease. So penetration now, the bacteria has mechanisms to cause penetration or to allow penetration of the bacteria into the host cell. So that is facilitated by the production and secretion of these invasions, which rearrange the actin microfilaments in the cell. And in this case, here we see the salmonella is actually causing this ruffling, so to speak, of the host cell membrane. And by doing that, it creates an opening, so to speak, for the salmonella to enter the cell. Now, other bacteria use slightly different mechanisms. We saw Shigella getting into the intestinal epithelium, and sometimes they can use the actin themselves to move into and through the host cell to travel to other adjacent cells. So bacteria can do damage actually to the host cells themselves. So once they overcome some of these defense mechanisms, then they can actually exert or create damage to the host cells. So they create damage by producing toxins. Now, these toxins, as you can imagine, can be very deadly and poisonous. So, and some of them certainly are, and they directly result in cell damage. These toxins can be transported by blood or lymph, and when these toxins become systemic, they can create a huge septic shock, for instance. These toxins can inhibit protein synthesis, and this is protein synthesis by the host cells. They can destroy blood cells and blood vessels, and they can disrupt the nervous system as well by causing spasms. So there are two major types of toxins. There's exotoxin and endotoxin. And in terms of the terminology utilized with toxins, we have, of course, toxin is the substance that contributes to the pathogenicity of that bacterial disease. So sometimes it's not the bacteria, it's the toxin. Toxogenicity is the ability to produce a toxin. Toxemia is the presence of the toxin in the blood, so how much toxin is in the blood. Toxoid is inactivated toxins that are used in vaccines, and then antitoxin are antibodies that are directed against a specific toxin. So again, there are two types. So the first type we're going to discuss is the exotoxin. And the exotoxin is toxins that are produced inside some of the bacteria, and these toxins are then secreted by the bacteria into the surrounding medium. So the exotoxin is released. Now, the exotoxin can sometimes just be released as the bacteria is part of the life cycle of the bacteria. But sometimes, and even more importantly, when this bacteria undergoes lysis, then we get a release of the exotoxin. And sometimes that can happen due to an immune reaction or to an antibiotic agent, for instance. So these exotoxins are proteins, they're enzymes, and they catalyze certain biochemical reactions, and often you don't need that much exotoxin to create these disease symptoms. So the bacteria that can produce exotoxin are gram-positive and or gram-negative. The exotoxins are soluble, and they can move in the blood and the lymph. They can travel in the blood or the lymph. And again, some of these diseases, they're not caused by the bacteria, they're caused by the toxins. And that the body can produce antibodies to these toxins. So in that respect, it's not a complete loss here. There are some defenses that the host can use to overcome these toxins. So some examples. Now, these are pretty serious disease. So diphtheria produces toxins. Is erythrogenic. Streptococcus pyogenes produces exotoxins. We have botulism. We have tetanus. We have Vibrio. Remember Vibrio cholera. We have staphylococcal enterotoxins. The Staph aureus creates toxins as well. So these are pretty serious diseases, and they're all caused by toxins. Now, here's just an example of one of them. They all have their various versions and forms, but here we actually have a binding and an active toxin. And so if you can see here in the figure, we have an A and B. So A is the active part of the toxin, and B is the part that facilitates binding. So we have the toxin released by the bacterium. We have binding. We have endocytosis here, and both of them actually come into the cell. Eventually B will leave and A stays behind, and then A it alters cell function by inhibiting protein synthesis. And then the B, can certainly go off and join another A and then certainly continue with the whole cycle. But again, this is an exotoxin. It's not a bacterium. It's a product of bacteria. Now, the endotoxin. Okay, and the endotoxin is different from an exotoxin. Now, endotoxin is always produced by gram-negative cells. So gram-negative bacteria, endotoxins are associated. They're on the outer portion of the cell wall. Now, remember, the cell wall of the gram-negative bacteria are that they have that outer membrane. So that outer membrane is partially composed, so the lipid portion of the outer membrane is potentially capable of being an endotoxin. So, and if you recall that lecture that we had on membranes, that lipid A was part of the gram-negative cell wall on that outer membrane. So these endotoxins are not proteins and enzymes. They're remember lipid polysaccharides. So they're lipids joined to carbohydrates. So when gram-negative bacteria undergo lysis, endotoxin is liberated. Okay, that's an important point. So antibiotics used to treat gram-negative bacterial infections can cause the endotoxin to be released, and sometimes the symptoms are worsened from the disease because of use of the antibiotic agent. And then fortunately, we can have ultimate recovery in some cases. So these endotoxins, they stimulate the host cell macrophages to secrete a high concentration of cytokines, and that inflammatory process is what causes or what is responsible for the deleterious effects from these endotoxins. So here is the endotoxins and the pyrogenic response. Now remember, pyrogenic refers to fever. So we have the bacterium with the endotoxin on the outside being engulfed by a macrophage, and this endotoxin on the outside causes the macrophage to secrete high amounts of cytokines, and these cytokines are what trigger inflammation as well as fever, because the cytokines travel to the hypothalamus, and it's the hypothalamus that causes it causes the metabolism of the body to increase, and then the temperature goes up. So the example endotoxins are pretty serious diseases also. So we have Salmonella typhi, which is the causative agent of typhoid fever. We have Neisseria meningitis, which is the causative agent of bacterial meningitis. And let me tell you, bacterial meningitis is worse than viral meningitis for sure. And then we have various bacteria that cause urinary tract infections. So the steps of a bacterial infection, to review, there requires entry into the host through a portal. Okay, sufficient numbers of the bacteria are required. The bacteria need to adhere to the host tissue. They need to colonize. They need to penetrate or invade the host defense mechanisms. That the host is going to launch an immune response, and the bacteria need to withstand that. The bacteria can then cause damage to the host tissue directly through direct infection or indirectly through toxins. And the host can either progress, or the bacteria can certainly survive, or the host can eradicate the bacteria and then fully recover. So as you can see, there are a lot of places where you can overcome the bacteria, but there are also a lot of places that the bacteria can overcome these various mechanisms that the host uses to defend itself. So in terms of the penetration, we saw that these bacteria had capsules, that there were certain components in the cell wall. We had enzymes, we had M proteins, we have fimbriae, we have that ruffling mechanism, that we had, depending on the variation of that cell wall, is there enough antigenic variation to create an immune response? And then we saw various ways in which that bacterium can actually penetrate into the host cell cytoskeleton. So our first example, we have the question, how do you think the strategies that bacteria overcome or employ to overcome host defenses relate to the tissue that bacteria infect? So we saw a huge variety of tissue, and we saw that one example, we had a capsule, right, that surrounded 2 diplococci, right? And those were gram-positive bacteria, but we had a capsule, and remember, having this capsule increased the virulence of that particular bacterial infection, and that was found with the pneumonia bacteria that lives in the respiratory tract. Now, you know, we have these places in the body, like the bladder and the stomach, right, where, I mean, it's almost by definition, potentially these bacteria cannot survive with oxygen. And they also need thick cell walls. They need, you know, bicarbonate in the cell wall, something to maintain the integrity of the bacteria in these very extreme environments. We have these various enzymes, the M protein, we have fimbriae, we have various ways, the tools that bacteria can use to enable itself to slow down, to hold on, so to speak, to get their way into the cell, to infect the cell, to release toxins that further destroy the tissue. So, you know, it's a wonder that we're all, you know, not suffering from bacterial infection all the time. I mean, it's pretty amazing. But, you know, if you listen to some of the lectures in the immune system, you will see that the human host has quite a few tools and defense mechanisms itself. So the whole interaction is quite complicated. But if, for instance, you're focusing on treatment or study of a particular bacteria, it's important to really get a good understanding. Where does it enter into the body? What kind of tissue does it live in or does it live with? You know, what are these mechanisms it utilizes, and can we find weaknesses in these mechanisms and develop agents to actually seek out those weaknesses and use those weaknesses against the bacteria? So the second question has to do with, what are the differences between an exotoxin and an endotoxin? Now, this is likely to be on, you know, an exam. For instance, it would be a great essay question. It would be great matching, multiple choice. So let's break it down. So remember, the exotoxin, okay, was a toxin produced by gram-positive and gram-negative, both, right? And an endotoxin was only by gram-negative, okay? Now, the exotoxin, remember, was secreted outside the bacteria. Oops. It's secreted outside the cell, okay, and it could stay inside the cell and be released during phagocytosis. Now, the endotoxin, remember, was part of that outer membrane, okay, and we had various parts of that membrane that contained what? Remember that lipid A, right? That lipid A was really important in relationship to it being a toxin. Now, it's the same thing. We can either, when that cell is engulfed by phagocytosis, remember the macrophage released all of those cytokines. So the endotoxin can actually cause direct tissue damage, and the cytokines and the endotoxin was more of an indirect. So it caused the macrophage to actually secrete the cytokines. So it was more of an indirect mechanism. So that's the way to tell the two apart, is just to kind of go through each property that each of them has and see how they are similar and also how they are different. Now, the last example has to do with what are the dangers of treating septic shock with an antibiotic agent. Number one question you have to ask. First of all, you have to find out what bacteria caused the septic shock. So what bacteria is responsible? Okay, number one. Now, if this is a bacteria that produces a toxin, we saw in both cases, we saw an endotoxin example and an exotoxin. What happens when you destroy the bacteria with an exotoxin? That bacteria releases all of it, right? With endotoxin, you can have, it can be released after destruction as well. So the point is, you've got to make sure that if you give antibiotic to somebody in septic shock, if it's a bacteria that produces a toxin, you better make sure that you figured it out enough so that you know that giving that patient an antibiotic will not actually kill them. I mean, that's like really important. So, what an interesting lecture. Thank you so much for visiting educator.com.